Good morning. Today we're going to do dihybrid crosses. Up until now, we have only looked at monohybrid genetic crosses where the inheritance of one characteristic was involved. For example, only the height in pea plants. Okay, with dihybrid crosses, we now deal with the inheritance of two characteristics or traits. Mendel conducted an experiment where he crossed pure breeding homozygous pea plants that have seeds that were round and yellow with pure breeding homozygous plants with seeds that were wrinkled and green. Okay, if we had to look at a picture there's the round and yellow seed crossed with another plant with green wrinkled seeds. In summary, what he then found was that all the offspring had round yellow seeds. He then took it a step further. Okay, he crossed two offspring with each other, okay, the F1 offspring. He then found that the F2 generation produced the following seed types: 9 round yellow seeds, 3 wrinkled yellow seeds, 3 round green seeds, 1 wrinkled green seed. Okay, you will see now with the dihybrid genetic crosses, we have a lot more possible combinations for the offspring. Right, so from this experiment now, Mendel came up with a new law. Okay, and that is the law of independent assortment, which states the various factors, okay, remember he called them factors. He didn't know they were alleles yet. That was a name that came up later. The various factors controlling the different characteristics, seed shape, seed color, plant height, etc., are separate entities, not influencing each other in any way and sorting themselves out independently during gamete formation. And therefore we have this huge variety. Okay, so now we're going to look at, if we go to page 82 in the textbook, we're now going to look at the F2 generation. Okay, or first, sorry, we're going to look at his experiment where he crossed round yellow seeds with wrinkled green seeds. Right, so firstly, okay, so here is the genetic cross for Mendel's first round of experiments, where he crossed the yellow round seed with the green wrinkled seed. Okay, we have a key there. R, capital R is round because it is dominant. Why? Because they told us that all the offspring had round seeds. Okay, therefore round's dominant, wrinkled is recessive. All offspring had yellow seeds, therefore yellow is dominant and green is recessive. Okay, so our phenotype, round yellow times wrinkled green. Okay, we were told they were both homozygous or pure breeding, so we will have, it has to be the same for round. We've got to have 2 capital R's and 2 capital Y's because it is homozygous. Same for the wrinkled and green. They must both be the same. Okay, then our gametes formed 2 capital R's, 2 capital Y's times that. Right, now let's have a look. Let's do a Punnett square. Okay, so now the Punnett square is slightly different because we have 2 characteristics in each block that are going to be crossed with each other. Okay, so if we look here again, right, remember we always do the first organism first. We will take these two letters, okay, this letter, and we will put it over here, and the second letter will go over here. The second organism will go down like that. Okay, so now we're crossing it. The R's will come first, okay, capital R again, and small r. Then capital Y, small y. Same here, R's first, capital R, small r, capital Y, small y. And if you look, they will all be the same. So that'll be your Punnett square for this one. Okay, going back here, our genotype, we will have 4 big R, small r, big Y, small y. So they will all be round and yellow. Okay, so now if we look at Mendel's second experiment where he crossbred 2 of the offspring, we will set it out like this. Okay, we have a key, and that key is capital R for round, small r for wrinkled, capital Y for yellow, and small y for green. Okay, we set out our genetic cross, but now this is the offspring. Okay, it's the second parent generation. So it will be P2 and down here F2. Also leave a bit of space here because we're going to have a lot of combinations for the genotype and the phenotype. Your Punnett square, you will do further down. It will be a lot bigger than the Punnett square that you are used to because there are so many more combinations, but you'll see in a moment. Okay, so firstly, we are crossing the offspring, the F1, first filial generation. They were all round and yellow, but they are heterozygous. Okay, and that will show in the genotype. Heterozygous for round, heterozygous for yellow. Remember, meiosis, we leave open our gametes. Okay, now fusion, we will do our Punnett square. Okay, so this one comes first, right? This first letter here, we will double it. Okay, so the R, capital R, small r, small r. Then we look at the Y. Okay, we're going to alternate it now. Capital Y, small y, capital Y, small y. Now you can see we have all the possible combinations of those 2 characteristics. Okay, then we do the second organism. We take the R, we double it. We take the small r, we double it. Then we take the Y, we alternate it. Capital Y, small y, capital Y, small y. Now we're going to cross it. Okay, we look at the R's first. There are 2 capital R's, 2 capital Y's. I like to go across. You can go down however you want to go as long as it is organized and work carefully. 2 capital R's, capital Y, small y. Capital R, small r. 2 capital Y's. Capital R, small r, capital Y, small y. Then we move on to the second one. Okay, so you can see this is going to take a bit of time. 2 capital R's, capital Y, small y. 2 capital R's, 2 small y's. Okay, so now your Punnett square is complete. Just check that it matches mine. Now we have to set it out in the genotype. This is why we need 2 lines. Okay, so we will start with this first one. Capital R, capital Y's. Now you have to take your pencil. I'm taking another color pen, and I'm going to tick how many of these there are in this very big Punnett square. So we've got 1. Okay, we look at all the capital R's. There's only 1. Okay, tick it off so you know it's been done. Then we go and look at the next one. Now I'm going to use a cross. Capital R with 2 different Y's. Go and look at my capital. There we go. There's another one. 2. Okay, there's only 2. Right, then I go to the next one. And now I will use a little dot. Okay, do the rest with me. We'll see the finish. Okay, so this is your final Punnett square. As I said, you need to use different symbols to mark off. You should have these results with the genotype. Okay, and then add them together to see there are 16 blocks. So 2, 4, 6, 8, 12, 13, 14, 15, 16. If you have 16, you know you've done it correctly, more or less. Then the phenotype, you will have 4 different types. Round yellow, round green, wrinkled yellow, wrinkled green. Okay, go back to your genotype. You should find that you will have, if you look at all of them, you should end up with 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Again, it should add up to 16.